This recorded lecture is the completion material for module number 8 on the nucleus, chromosomes, and DNA. So, we already covered the discussion topics on the nucleus. We also covered the biochemistry and the structure of the DNA. We discussed about um, what do you call this? We also discussed about the levels of DNA folding, especially in eukaryotic organisms. So for this completion lecture, we will still talk about some of the important features of the DNA as the primary informational molecule of cellular systems. Then afterwards, we will cover a comparison discussion on the DNA of eukaryotes and prokaryotes, and I will be introducing the three processes underlying the flow of information in molecular biology. So we'll have a brief discussion on DNA replication, transcription, and protein translation. Okay, so last time we stopped with the discussion on, I don't know, DNA supercoiling and the types of chromatin. So, aside from those things that I've already mentioned about that describe the different features of eukaryotic DNA, another characteristic is the capacity of DNA to denature and renature. Okay, so the ambient temperature dictates the conformity, the native conformation of a DNA molecule. A certain temperature called the melting temperature, T sub m, will be indicating the threshold for DNA, the native double helical DNA, to stay double helical. No, so if the ambient temperature breaches beyond the melting temperature threshold, the DNA double helix will denature. So the helical conformation will be gone. So you can expect that the anti-parallel strands will dissociate from one another. So the H bonds between the nitrogenous bases will be destroyed. Now, if the same DNA molecule that has been denatured is allowed to cool down, okay, so you expect that reformation of the DNA double helix is possible. Okay, so you expect that the anti-parallel strands can renature. Okay, so they will zip again. So then the original native DNA can exist after cooling the setup. No. Um, what else? Another important thing, another important note I want to share with you guys is this feature of the DNA, its capacity to denature and renature, has been utilized by techniques such as polymerase chain reaction. No? So in PCR, the DNA molecule is subjected to cycles of denaturation and renaturation in order to amplify a segment, amplify a gene or a biomarker within the nucleic acid molecule. So it takes advantage of the capacity of the DNA, or the characteristic of DNA, to denature and renature. Okay. Now, in addition, for particularly for eukaryotes, no, we introduced the idea that um, a portion or certain portions of the eukaryotic chromosome is actually comprised of what do you call this, non-coding or repetitive sequences. No, so the chromosome centromere, which is known as the primary constriction, diba? This area of the chromosome is where your sister chromatids are connected. It's where they're attached, and there are several positions for the centromere sequences. No, there are centromeres that can be found at the middle of sister chromatids. We call that metacentric uh, positions that are beyond the or I know not really in the center. So we can call that the submetacentric position. There are also centromeres on the tips, no, of the sister chromatids. Tinatawag natin na acrocentric or telocentric. Okay. So here you can see the set of human chromosomes, and you can see the primary constriction indicated by the centromere. So more about the centromere. Okay. So during cell division, we expect these segments of DNA to be very functional. Okay, kinetochores attach to it serves as spindle fiber attachment locations, and as important parts of the metaphase chromosome of a dividing cell. Okay, the area of the centromeric area or the centromere repeats are composed of short tandem repeated sequences. So they're condensed and they're constitutively heterochromatic. So they are not accessible to elements of replication and transcription. Okay, so these repeat sequences they can range from between 5 to 170 base pairs. Okay, so as an example, we have the human alphoid satellite repeat that can serve as centromeric sequences. No? Now, among living organisms, we cannot really identify, or there's no identifiable universal centromere repeat. Okay, but within species or between species, these repeats can be found. They can be found at the same frequency. Okay, they're also highly mutable. They can mutate. They can be they can possess mutations, single base mutations, and what do you call this? These since they're non-coding, it's not really something to consider as problematic, no? Kasi kung dahil non-coding naman siya and essentially just acts as a attachment of some sort or constriction of some sort within chromosomes, then there's no problem dapat, no, kapag kahit na may mutation within the centromeric sequences. Okay. There are proteins also that can be associated to these centromere sequences. So we call them centromere specific histone proteins. No? What else? Here, let's take a look at a model of the centromere. So here you can see a central centromere, a metacentric centromere. Okay, so you can appreciate that the repeats are highly are comprising the ano no the satellite DNA monomer. Okay, so and then these highly ordered repeats, and they of course exist at particular frequencies, no, and they can a specific range or number of nucleotide pairs are expected to be characteristic of that highly orderly repeat, no, for the centromere sequences. Here we can see a centromere. Okay, so you can appreciate the kinetochore as an area for spindle fiber attachment. You have your spindle microtubules attaching to it. Okay, then Particularly, itong constriction na to is heterochromatic, no? Formed by satellite DNA, by uh, our highly orderly repeats of the centromere sequences. Okay. So yeah, we're done with centromeres, with DNA being capable of denaturing and renaturing. Another important feature of cellular DNA is its variety or diversity. No, so something that we can appreciate from this illustration is the fact that not all individuals under the same species will possess the same profile all the time. So it's not going to be 100% similar among individuals under the same species. So we have this so-called single nucleotide polymorphisms, which are variations of a single position in a DNA sequence. So um, SNPs, as we often call them, are associated to both coding and non-coding DNA. So they can be found in genes that can be translated. They can also be found in non-coding sequences. And in terms of their function for analysis, they're highly stable sources of information, especially for doing lineage tracing for a particular organism. SNPs are very useful as a bioinformatic marker. Also, they can be used for molecular phylogeny analysis. You can extract the SNPs of a DNA sequence or of a gene for a particular organism, and then use that information to create a genetic tree, no, a gene tree. So uh, the SNPs are may produce a consequence or not. It's possible na, especially kung nasa coding sequence siya, no? So the SNP can alter the conformation of the protein that is encoded by the gene. Okay, so as you can see here, a variant of the genotype from the common or from the wild type is considered to be a SNP, no? And in some cases, it's possible that the protein will be altered because of that single base change or that single base variation. Okay, so yan, we're good with another feature of eukaryotic DNA, which is single nucleotide polymorphisms. Actually, let me qualify now. Not only eukaryotic, but also for prokaryotic DNA. Now, I think the last, or not really the last, but another feature that I also want to talk about is the fact that eukaryotic genomes are really large. So in our lecture, I emphasize the one difference between eukaryotic and prokaryotic DNA. Eukaryotic DNA you expect to be very large in terms of size, while prokaryotic less than 9 MB, most prokaryotes will have a total genome size of less than 9 megabases. Okay, so for eukaryotes, it will be millions of bases. Okay, and the reason behind the observation that eukaryotic genomes are really large, are appreciably large, is the fact that there are what we call repetitive DNA sequences. So the centromere is one example, no? But aside from it, there are a number of subtypes under the repetitive DNA sequence family. So there is what we call tandem repeats, like the ones I mentioned, the microsatellites, mini satellites, and then other non-coding DNA sequences. And we also have dispersed repeats, which are associated with DNA sequences that move from one location to another. Meaning these DNA sequences, they are not conserved at a particular position of the entire genome. It's possible that at one point they're found at a particular location, let's say 900 to 950, and then after some time it's found in another place. So ito yung mga tinatawag natin na transposons or jumping genes. Actually, the word jumping gene or the qualifier, the description jumping gene is quite it's quite inaccurate kasi not all jumping DNA sequences are considered genes, are considered sequences that code for a protein. So let's just call them jumping DNA sequences, no, mga transposons. That do not have a permanent location in the organism's genome. Okay, uh, the repetitive DNA sequences are very high in terms of diversity. Now, within individuals under the same species, diverse tandem repeats, diverse repetitive DNA repeats can be found. Okay, and the fact that these repeats are amplified and deleted through time actually contributed to the wide variation in terms of the genome size of different eukaryotic organisms. No? In the animal kingdom, the genome size could vary from between 20 megabases to 130 gigabases. And primarily, you can attribute that to substantial content of repetitive sequences. Okay, for plants, same case, no? The repetitive genomic DNA comprise a quarter, probably no, siguro 10 to 25% of the DNA of the plant genome is actually comprised of repetitive DNA sequences. Okay, so we're done with all the features that I want to I want to talk about sa DNA. So we've enumerated those important characteristics of DNA. We've discussed them. I've given you an overview of each of these characteristics. Now, how do we know, or why do we know so much about DNA and its features? Of course, we have technologies such as DNA sequencing. So a lot of the things that we know about DNA of eukaryotic organisms and prokaryotic organisms are hinged dun sa sa discovery of a technology where you can or we are able to sequence DNA segments or the whole genome of a specific organism. Okay, so before we have this what we call Sanger sequencing, where we can sequence a DNA segment using a termination technique. Now we have high throughput sequencing, so called next generation sequencing. So in next generation sequencing, there is multiple massive parallel sequencing happening. So kung sa Sanger sequencing pa isa-isa yung molecule that's being sequenced, under NGS, under high throughput sequencing, imagine you have a panel of you have a panel of detection. I don't know, detection. You have a detection panel with multiple wells, and each well can cater to, or each well can cater to a specific molecule. Kaya parallel yung sequencing, no? Kaya tinawag siya na parallel. So we have to give it to inventors and companies, private companies that keep on innovating such that our capacity to, or our knowledge base about DNA in its characteristics is actually increasing over time. Okay, now since I've talked about, I introduced the idea of DNA sequencing to this lecture, let me also introduce another important concept: genome. No, so when we talk about the DNA found in a single cell, all the DNA. When we talk about all the DNA in a single cell, meaning the DNA found in the nucleus, the DNA found in, let's say, the mitochondria or the chloroplast if you have a plant cell. Okay, all those DNA, they comprise what we call the total genome or the genome of that cell. Okay, and it has been observed that genome size is actually related to complexity of the cell of the living system. No, your simple organisms, your unicellular organisms, bacteria, your uh, unicellular fungi, and your viruses, they have smaller genome, total genome size compared to the animals or compared to the plants. No, so in a way, we can say that genome size is related to it's related to how late the organism appeared in the evolutionary timeline. I cannot I actually I refuse to no no I refuse to say that genome size is really related to the complexity of the organism because there's no benchmark to really say that a unicellular organism is less complex compared to, let's say, a plant. We can't really totally compare an apple with an orange because they're different systems, diba? So I would like to qualify for this slide, no? We can say na the size of a genome is related to is related to the timeline in which the organism appeared, no, in our evolutionary clock. So the earlier ones, the of course the unicellulars, yan sila mas maliit yung kanilang genome, no, compared to the later appearing organisms like plants and animals have bigger genome sizes. Okay, so uh, yan. Okay, so I qualified already my statement for this. What else? Ito, some of the common things that you will encounter as you look into genomics, and you will encounter some abbreviations like the ones that are used to describe genome size. We have KB, kilobases, that's 1,000 base pairs. That's equivalent to 1 kilobase. 1 million base pairs, 1 MB is equivalent to 1 million base pairs is equivalent to 1 MB. And 1 billion base pairs is equivalent to 1 GB. So, for example, if you have an E. coli cell with around 4,639,221 base pairs, you can express it in megabases as 4.6 MB. Yeah. So for bacteria, you expect very small genome sizes, unlike humans. Okay. I have here an interesting resource that I'm sharing to the class. Okay, so you can go to the website of NCBI here. Yeah, so actually this one. You can go to NCBI's website and then look at use the genome database. If you click on that, and then you write the scientific name of the organism that you are looking for, you will be directed to a page. You will be directed to a summary page that actually provide current information with regards to genome sequence of the organism that you searched for. So, for example, for the case of Homo sapiens, for us, we can see here in this summary page yung chromosomes, the what do you call this? The ideogram. So ito yung ideogram chromosomes natin. And then these are the reference sequence accession codes. Like if you click on one, for example, for chromosome number 1, this is a reference sequence accession number. Ito siya, no? Ito yung kanyang, these are the authors who did the sequencing, and then hindi natin actually makikita yung data. It's very big. So the data is encoded, or the data comes into a redacted format. And I won't go into the details of that. I'm just introducing to you the fact na NCBI really holds a lot of information with regards to DNA sequences and genomic sequences. Okay, so Other examples, sabihin natin, let's have here Escherichia coli. Escherichia coli. If you look for Escherichia coli under the genome database, ito yung mga available. No? And then what are we observing? We're seeing that for bacteria, we don't have a chromosome number. Yeah, so important thing, okay? Bacteria are non-multichromosomal. Okay? Isa lang ang chromosome ng bacteria. Most of bacterial species. Though there are certain bacterial species na ang convention is parang kinoconsider na dalawa yung kanilang chromosome. But all bacteria or majority of the bacteria that we know and that we have studied are all possessing a single chromosome. Okay? So let's take a look at this one as a reference. Ito. Yung chromosome ng E. coli K12 substrain, which is a common E. coli strain. Yeah. Okay. So you can see here information with regards to ako sino yung nag-sequence, and then paano natin makukuha, or the related journal where the sequence is used, no, as a evidence material, as evidentiary material. Okay, now looking at the chromosomes, ayan. Here, I think this is chromosome number 1 of the human animal, the human species. So here's the ideogram, and you can appreciate yung annotations for a particular part or a particular component of the chromosome. Kung ano yung makikita for particular area. Yan. Okay. I won't go into the specifics of this one. Hindi pa naman nyo kailangan talaga malaman ito. But I just wanted to introduce yung information that is already available, or the databases that are useful for accessing information like genome sequences of living organisms. Now, moving forward, so done with this one. Let's take a look at, or let's remember that it's not only nucleus that contains DNA. In eukaryotic organisms, some organelles also possess their own DNA. I've already discussed this a long time ago. So we have the mitochondria and the chloroplast as organelles that possess their own genetic material, their own DNA. For the case of the mitochondria, here you can see the genome of the human mitochondrion, no? Which is composed of about 16,500 plus base pairs. And as a circular DNA, no, similar with bacteria, no, with prokaryotes, circular ang DNA ng mitochondria. Certain segments code for particular parts or particular products, particular protein products. For especially those important for the electron transport chain. Okay. Now, we're now at the juncture of our lecture where we compare prokaryotes and eukaryotes in terms of their chromosomal material, in terms of their DNA material. So I've mentioned chromosomes of prokaryotes are single, and usually they, in some species, they're accompanied with plasmids. So plasmids are also made up of nucleic acids, no? But they're not technically consequential compared to the chromosomes of prokaryotes. No? While eukaryotes are composed of multiple chromosomes. In terms of the structure, prokaryotes possess a circular DNA, a circular chromosome. So the ends actually meet. While eukaryotes are linear, so ang chromosomes natin are actually not going to, the ends of our chromosomes, they do not meet. They don't form a circle, unlike that of bacteria or archaea. For prokaryotes, their chromosomes are made up of nucleic acids, purely nucleic acids, while for eukaryotes, as I've discussed in our synchronous session last week, or this week, eukaryotes, we can call the chromosomes nucleoproteins because aside from nucleic acid sequences, meron ding histone proteins, no? May proteins na kasama. For prokaryotes do not have a nucleus, therefore their chromosomes or DNA material is just suspended in their cytoplasm. While for eukaryotes, we have our nucleus. So it's our chromosomes are suspended or are embedded in the nucleoplasm. Okay. In terms of what happens during division, prokaryotes copy their chromosomes and divide immediately afterwards. While for eukaryotes, there is copying of chromosomes. The cell follows a period of growth, and then the cell will enter the period of division. It will enter the period of mitosis, such that the doubled or copied chromosomes will now be submitted to the daughter cells, to the I don't know, the progeny cells. Okay. Now, sa primary genetic material, the major characteristics of DNA can now be summarized by these by these articulations or these characteristics. Okay, so we were now relating not only the structure but also the functions and the characteristics that are important for functions to take place, or for the function of the DNA molecule to be kept, no, or to still to be maintained. Okay, so the DNA molecule must be able to duplicate itself with extraordinary fidelity. So there should be very high integrity in terms of DNA duplication. The molecular structure then must be very stable. Mutations may happen, but they should be at very low frequency. So, aside from the debate about DNA and protein, DNA versus proteins, yung napag-usapan natin, no, about primary genetic the debate about the primary informational molecule of living systems. Currently, I would say, no, very current lang, that there are some individuals who posit the idea of an RNA world first prior to a DNA world. Na they hypothesize, they give us the idea na initially daw, it was RNA that served as the primary informational molecule. Tas afterwards, nagkaroon ng shift, nagkaroon ng mas mataas na fitness ang DNA. So, mas naging ano siya, mas naging capable siya of actually carrying the information in cellular systems. So, nasabi ko yan or na-include yan sa discussion because unlike RNA, DNA is more stable in terms of mutations. Mas prone ang RNA sa mutation, especially that most of the time it exists as single-stranded in nature. Okay. Now, also another factor, another characteristic for DNA as a genetic material is whenever there is an error of replication, whenever there is mutation, that error is expected to be faithfully duplicated as well. Ba? Kaya nga, inborn or congenital, ano yan? Diseases that relate to mutations, talaga sila. They can really be submitted from one generation to another because the disease, which is a result of error of division, error of replication or mutation, is faithfully submitted from the parent to the offspring or from the parent to the progeny. Okay. Also, we without a doubt, the DNA as the primary genetic material is capable of holding all necessary biological information. And aside from holding it, the DNA must be able to transmit the information from generation to generation. And that is beautifully done using additional molecules, using the RNA to be specific. No? Kasi holding the primary information, okay, it's very important that the DNA preserves or that the DNA is really protected, okay, from activities. Kaya magkaroon tayo ng ano, no? Kaya in a way we can relate replication and transcription to the inherent need to really protect DNA because it holds all biological information in living systems. Now, the process of the key processes under central dogma, which are the processes of replication, transcription, and translation, we can conceptualize them using a lens of information flow. Yeah, using the information flow lens. So you can think of proteins as outputs, and you can think of DNA sequences as inputs. And the journey of DNA sequences being decoded, transcripted, and later on translated as protein molecules. That journey is an outstanding example of information flow within cells. To be specific, lateral information flow. Lateral kasi only occurring at a particular cell, at a one generation of cell. No? But if you are to look at it naman in the under the replication or under the lens of inheritance, pwede rin naman magkaroon ng horizontal, no? Or rather vertical. So lateral or horizontal flow of information. And we also have vertical flow of information. So from your parent cell, right? The DNA code is faithfully replicated, submitted to the progeny cells, and essentially whatever the code of the parent will be the code of the of the progeny cells. So ang tawag naman natin doon, vertical flow, no? Vertical flow of information. Okay, I will before before I discuss before we talk about the an overview on the processes of DNA replication, transcription, and protein translation. There are a few interesting stuff that I'd like to share. So first is uh, in relation to information flow, no, the area of the bacteria which is dedicated to concentrating nuclear material. So this area is called the bacterial nucleoid. It's a space that's not enclosed by a membranous material, and ano siya, no? Dedicated area talaga siya within the cytoplasm of the bacteria. And um, we characterize it as being proximal to the plasma membrane. So it's somehow connected to the plasma membrane. Okay, and as an important space, it's really dedicated to ensuring that the prokaryotic DNA is doing its thing. The prokaryotic DNA is replicating, transcripting, and eventually be decoded as a protein sequence, as a protein code, no, as a protein sequence information. Okay. Now, aside from the bacterial nucleoid, another element that we relate to information flow in prokaryotes is the plasmids. So plasmids, as I've told you, are not chromosomal material. Okay, hindi sila major genome or part ng major genome ng bacteria. There are additional material, additional DNA sequences that can carry genes with them, usually virulence genes. And these plasmids are activated during stressful times. So ano sila, nadi-decode sila once that the bacteria or the prokaryote is subjected to multiple stressors. Okay, and once decoded, they can produce an output, a protein output. Okay. Also, another curious thing I'd like to, or interesting stuff I'd like to share. You'd remember yung satellites, no? Centromeres. Okay, so we have a technique called DNA fingerprinting in order to compare the profile of DNA from different sample sources. Okay, so as an example, a DNA microsatellite is very useful for comparing genomes from multiple individuals. Like for cases of, I don't know, criminal cases, DNA evidence is very useful in comparing or finding out if mayroon bang trace material that can be attributed the I don't know, to the perpetrator or the accused, no? Na usually kapag meron, di ba, that is a very strong evidence. Though it's still circumstantial, kasi it's possible that there is no link, di ba? Hindi naman natin 'yon may establish ng ganun-ganun lang. Not ano no, hindi lang presence ng DNA material ang magpo-prove ng guilt ng isang accused, no, for criminal cases where biomaterial are used as evidence, no. Now, going back, yun nga, very useful ang DNA for fingerprinting for uh, investigating criminal cases. Okay. In medicine naman, we have another interesting concept, yung transgenesis or a formation of transgenic animal. No? So in forming or creating a transgenic animal, you make use of genes that are responsible for a particular trait or a particular disease. You extract that gene, and then you introduce that gene to a fertilized egg, no, of an animal or another organism, where you can expect that gene to be expressed. Afterwards, embryos that are formed containing that exogenous gene will be implanted will be implanted in the uterus of a surrogate mother. Okay, the offspring, the produced organism, may or may not express the selected gene, and essentially, with that, we can do tracing of diseases, especially genetic diseases. So for genetic diseases, where we are already able to identify which genes are responsible for the disease, pwede natin gawin na the routine can be, you can trace the progress of a disease in model animals by creating transgenic mice, by creating transgenic model mice or model mammalian systems. Tapos makikita mo how we can trace how a disease can progress using transgenesis. Okay. Lastly, technique, no, a curious technique that I'd like to share. We have what we call fluorescence in situ hybridization or FISH, no, where you use probes that recognize particular portions of your chromosomes, and these probes, they can attach to they can attach to those particular sequences, and in a way you can trace anong nangyayari, no, kung ano yung complement, actually not necessarily anong nangyayari, but more on what sequences are complementary with one another. You can do that using this technique. So I introduced those three things because they are evident applications of our more than a century, almost magdadalawang century na, no, na understanding about the features of the DNA in cellular systems. I introduced those three things because these are are very obvious applications. Okay, I'm now at the last leg of my recorded lecture. We'll talk about the central dogma of molecular biology. We'll be talking about three important processes: replication, transcription, and translation. Okay, let's begin by looking at replication, DNA replication. Okay, so you remember I talked about, or I mentioned na yung sa publication ni Watson and Crick, they also propose a method, or not method, but a mechanism for the replication of DNA, no? And before, there were numerous hypotheses about DNA replication na hindi na that were already debunked, but just for the sake of course, discussing those things, yeah, there are a number of possible models for DNA replication. There's what we call the conservative mechanism, the conservative replication, where an original DNA double helix will yield a progeny, a daughter helix, which is entirely different from it in terms of which is entirely separate from it. Meaning that they don't share anything, any biomolecular material, any atom at all. So they're two separate molecules. Then we also have the dispersive mechanism or the dispersive approach. Yan. So pache-pache na the daughter the daughter DNA double helix will mix segments of it with the parent. So that's called dispersive. And we also have what we call the semi-conservative replication, which is the mechanism proven to be followed by cellular systems in replicating their DNA. So in semi-conservative replication, a parent double helical DNA will separate its antiparallel strands and will produce a daughter strand that will be related with the parent strand. So yung dalawang DNA molecule that will be byproduct of a single DNA molecule is composed of an old strand and a newly synthesized complementary strand. So may dalawa kang separate molecules na yun. Okay? So remember, this three mechanisms. But of course, we're only paying attention with the semi-conservative, where each daughter molecule will be composed of, again, one conserved strand from the parental molecule and one newly synthesized strand complementary to the parent strand. Okay. In the succeeding module, we will talk about the specifics of the process of DNA replication. But let me introduce to you what elements or what proteins are required for replication to take place. So, aside from proteins, no, what other elements? So one of the first elements that is required is, of course, the template DNA, the parental DNA, which serves as the master copy of the sequence or the gene. Also, there will be a requirement of deoxyribonucleoside triphosphates, DNTPs, which act as substrates during the process of polymerization in DNA replication. Another protein or another element is a protein, the helix unwinding protein, which unwinds the parental strands to create separate template strands. Another protein is the helix destabilizing protein, which are also called single-stranded binding proteins. So these the helix destabilizing protein, what they do is they attach to the separated template strands, and they prevent the template strands from renaturing or re-annealing with one another. Excuse me. Another element is DNA gyrase. Yeah. So we have DNA gyrase, which is helix helix relaxing protein. Wait lang I'll just get some water. Okay, so going back. Another element, another protein, we have the helix relaxing protein, DNA gyrase. DNA gyrase releases tension in supercoiled twisted DNA by unwinding the parental DNA without rotating the molecule. So, nag-unwind and not necessarily doing spatial rearrangement of the molecule. And of course, the very important enzyme, polymerase. DNA polymerase catalyzing the synthesis of the daughter DNA. And as an enzyme, it will require a 3' hydroxyl N as a primer for the addition of the nucleotides. Also, the process of replication involves RNA polymerases, responsible for synthesizing RNA primer strands. So I will discuss the importance of all these elements during our yung module na natin for central dogma. Another element, we have exonucleases that can run from 5' to 3' or 3' to 5'. This is 5, dapat 5 yan. Sorry. Okay, and these exonucleases cleave RNA primer from the elongation. So, nangyayari kasi, one of the segments or one of the daughter strands is called a lagging strand. So, for the lagging strand, yung synthesis niya is not continuous. It will require primers. So, that's where RNA polymerase actually comes into play. Then we also have, lastly, yung tinatawag natin na ligase enzyme. So the DNA ligase is responsible for joining the nicks and gaps, and they do that by catalyzing the formation of phosphodiester bonds in our daughter or complementary strand. Okay, so this is a nice illustration on what happens during DNA replication. So one strand is said to be the leading, and another is a lagging, where primers are required. Okay, I will be, don't worry, because I will discuss in depth yung process of replication, and we will look into the comparison and contrast of replication in prokaryotic and eukaryotic organisms. Now let's move on to our second step, or the second process, transcription. Yeah, so transcription, which is divided further into three sub-processes. There's a process of initiation, afterwards the elongation, and termination. So in transcription, you expect that your DNA is transcribed to RNA. And the RNA outputs are of different types. Generally, we have three types of RNA outputs. We have the messenger RNA, which has a size of about 300 to 12,000 nucleotides. They code the sequence for amino acids or proteins, and they form secondary structures. They form hairpin loops, yung messenger RNA. While transfer RNA forms tertiary structures. Unlike messenger, mas maikli ang transfer RNA, usually less than 100 nucleotides. They form a tertiary structure, the clover leaf structure that you can see here, possessing the anticodon arm, tong tatlong nucleotides na to, that is complementary to the codons found in messenger RNA. And you can expect that the profile or the nature of the anticodon will dictate the kind of amino acid residue attached to the transfer RNA. Okay? Then lastly, we have ribosomal RNA. No? I discussed this sa first part ng lecture natin, na within, di ba, in the nucleus, the nucleolus serves as an active site for the biosynthesis of ribosomal RNA. Okay, and as an important nucleic acid, it can come into different forms. There is a light type, a medium type, and a heavy isotype. No? And the ribosomes, they are comprised of they are complexes of ribosomal RNA and ribosomal proteins. Okay. Now, last topic, last 3 slides na lang ito for this recorded lecture. We have translation. Ito na, no? Yung production of the polypeptide, the production of the protein. Okay, so your mRNA, which is translated by the translational machinery by the ribosomes, your transfer RNA comes into play as it introduce, because it's important for the introduction of the amino acids. No? Now, an overview of the process. It's composed of three sub processes. An initiation step where the components of the translational apparatus will come together with a molecule of mRNA. So the translational apparatus comprised of the ribosome, and of course yung mga incoming tRNAs with their amino acids. Tapos that translational apparatus hinges or finds an mRNA to translate. Okay. Then, in initiation, a transfer RNA carrying the first amino acid binds to the start codon. And then, second process, elongation. Amino acids are now conveyed to the mRNA by the tRNAs coming in, and they are added one by one. So the polypeptide chain, the nascent polypeptide chain, will elongate one amino acid at a time. And then lastly, once that the translational complex recognizes a stop codon in the mRNA, the an element will come into to play, yung mga release factors, and they will disassemble the translational apparatus, releasing the synthesized polypeptide. Okay? So here's a very nice illustration kung ano ang nangyayari. So your initiation where your ribosome, mRNA, tRNA comes into play. Your first tRNA, no, recognizing the start codon, elongation. Then once that a stop codon is recognized, release factors will be involved, and the complex will be disbanded. Okay, the complex will be disbanded, and they will now be ready to be recycled. They can process another mRNA molecule. Okay, so don't worry, kasi we will be doing a deep dive into the three processes. We'll deep dive replication, transcription, and translation for both eukaryotic and prokaryotic systems. So, and mind you, this is really going to be ano siya, term filled, no? Maraming concepts or terminologies that we have to memorize. We have, I'd say memorize, because eventually you will be familiar with something that you know on top of your head. So important na ma-memorize natin so that you can easily recall them in the future. Okay? So that ends the lecture discussion for module number 8. Okay?